primordial follicles. Female reproductive success is highly depending on female age, and this is a phenomenon that we call ovarian aging. And to give you some examples of that, I have two graphs here. The left graph shows the percentage of pregnancies per ART cycle for women receiving their own oocytes using ART treatment or donor oocytes. On the x-axis, you see female age. And if you look at the continuous line, so those are the women receiving their own oocytes, you see that with increasing female age, the chances of becoming pregnant per ART cycle decline. While when these same women receive donor oocytes of relatively young donors, this whole age effect diminishes. Well, the same goes for the chances of suffering a pregnancy loss per pregnancy. Again, here you see on the x-axis female age, and on the y-axis this time you see the chances of suffering a spontaneous pregnancy loss per pregnancy. Again, these are women using ART treatment, and the continuous line represents women that receive back their own oocytes, while the dotted line represents women receiving back donor oocytes. And here again you see that after the age of around 35, there's a rapid increase in chances of suffering a pregnancy loss for women receiving their own oocytes, while when they receive donor oocytes, this whole age effect is gone. So we are wondering, what's the difference between an oocyte of an older woman compared to a younger woman? So actually, how does the oocyte quality decline with increasing female age? Most studies that focus on ovarian aging use oocytes after treatment, so oocytes after ovulation. So those are represented here in the cartoon. But we were wondering about the primordial follicles. Those are the oocytes that you see here. The primordial follicles reside in the ovary until they are being stimulated for ovulation, but this can take more than 40 years. During this time, they are exposed to a lot of environmental factors, like for example, reactive oxygen species. Those are oxygen molecules that can easily react to other molecules and by that cause damage. So during this time, when they are exposed to a lot of ROS damage, this could decline the oocyte quality. So our hypothesis is that the decrease in oocyte quality is a result of reactive oxygen species, or in short ROS, induced damage in primordial follicles. To examine this, we collected ovarian biopsies that were previously taken out during surgery. And we collected biopsies of women of different age groups that represent the oocyte quality decline over the years. So our youngest age group was women between 18 till 29 years of age, then 30 till 34 years of age, 35 till 39 years of age, and 40 till 45 years of age. The biopsies were embedded in paraffin, and the amount of blocks that we collected per age groups are represented here in orange, and it's about 10 per age group. So we received the biopsies, but then we were wondering, are there follicles present in the biopsies? So for that, we did a hematotoxylin eosin staining to identify the different follicles and the different stages of folliculogenesis. And I have a picture of that later. When we identified the follicles, we were of course wondering, can we find ROS-induced damage? And we were particularly interested in the primordial follicles. ROS can damage different intracellular compartments like proteins, lipids, and DNA. So we stained the ROS-induced damage for these different intracellular compartments with the antibodies that are presented here. When we found ROS, we were wondering where does the ROS come from? And the most known way of producing ROS is by using mitochondria. So for that, we wanted to stain mitochondria, but not only stain if they were there, but also if the follicles were able to use them. And for that, we stained pyruvate dehydrogenase. Pyruvate dehydrogenase changes pyruvate into acetyl-CoA, after which it goes into the citric acid cycle and can create ROS damage. So we stained pyruvate dehydrogenase to find out if the follicles were already able to use oxidative phosphorylation. This is a picture of the different follicles that we identified in our biopsies, the different stages of folliculogenesis. So like I said before, we were mostly interested in the primordial follicle stage. Primordial follicles are recognized by an oocyte surrounded by a single layer of granulosa cells that are flattened. When the primordial follicle is stimulated, the granulosa cells become more cubical. And this is what we call a primary follicle. When there's different layers of granulosa cells, we call this a secondary follicle. And the last stage of folliculogenesis that we could identify in our biopsies was the tertiary follicle. Here, there's a fluid-filled cavity that starts to exist between the different layers of granulosa cells. So when we found out that we could find follicles in our biopsies, we went back to the primordial follicles and wanted to identify if there was ROS-induced damage there. So these are pictures of the immunofluorescence that we did. And in blue you see the DAPI staining, which stains the nuclei. And you see here in the middle the oocyte nucleus. And the oocyte nucleus is surrounded by the nuclei of the granulosa cells. In red, you see the ROS-induced damage staining for proteins, lipids, and DNA. So here you can see that there's protein, lipid, and DNA-induced ROS damage present in the primordial follicles. But we were wondering, okay, if it's present, does it also increase with increasing female age? And for that, we measured the peak intensity of the antibody that was present here for the different age groups that we used. So here you see a graph of the different age groups and the peak intensity of the antibody that we used for protein, lipid, and DNA damage. And you see that the protein and lipid damage increases with increasing female age, while the DNA damage does not seem to change. So we hypothesize that DNA is somehow protected from ROS-induced damage. The increase in the protein and lipid damage was about 50%. So after we identified ROS-induced damage, we wanted to know where the ROS-induced damage was coming from. And for that, we went back to all different stages of folliculogenesis and stained if there were mitochondria present and if there was pyruvate dehydrogenase present. Here again you see in blue the DAPI staining, green borders the oocyte from the granulosa cells, and in yellow you see the pyruvate dehydrogenase, and in red the mitochondrial staining. If you zoom in intensively, you will see that all the yellow is co-stained with red. So if you see here only yellow, this is actually a co-staining of mitochondria and pyruvate dehydrogenase. So we found out that during all stages of folliculogenesis, mitochondria were present, and there was also pyruvate dehydrogenase present, which gives them the ability to use oxidative phosphorylation. So to conclude, we have showed that primordial follicles already show reactive oxygen species, or in short ROS-induced damage, and that the ROS-induced damage to proteins and lipids seems to increase with increasing female age. And we found out that the ROS could come from the oxidative phosphorylation because there was pyruvate dehydrogenase present in the primordial follicles. And actually this was present throughout all stages of folliculogenesis. The technique that we used here is very limited for statistics, but I think that we showed that future studies should not only focus on the oocyte right after ovulation, but also focus on the primordial follicles, especially in regard to ROS and oxidative phosphorylation activity. I would like to thank my colleagues at the Center for Reproductive Medicine, at the Laboratory for Genetic Metabolic Diseases, Human Movement Sciences, and the Department of Pathology. And I would like to thank you for your attention.